Hi everyone, welcome to the ASIG meeting for this month. Please introduce yourselves in the chat here. I'm just going to wait a few minutes because we've got over 100 people booked on this event and currently we've got 30 of you. So we'll just wait for a few more people to come in. Yeah, I'm just admitting people into the waiting room. Somebody's got their microphone on. Helpful if you could turn your microphone off. I'll just give it a couple of minutes. Okay, well, we've got over 30 in now. We've got somebody else with a microphone on. Okay, yes, I'm going to stop sharing my slides. If you'd like to start sharing yours, and I'll introduce you.
Will do.
Okay, we've got Professor Richard, known as Delahay, speaking this evening. He's been involved with scientific research as a wildlife biologist since 1990 and in ecological consultancy since 2009. He's got a long career as a research scientist working on wildlife management and disease ecology and government research agencies, and is visiting professor at Exeter University. He's published over 160 scientific papers and book chapters on various aspects of wildlife biology, including the management of human-wildlife conflict and the ecology of wildlife diseases. He's also director of the ecological consultancy RSK Biosensors, where he provides technical advice across a range of projects and has responsibility for research and development in ecological techniques. Over to you, Des. And if anyone has any questions, please put them in the chat and we'll deal with them when Des has finished speaking.
Great, thanks very much, Debbie. I'm going to turn off my my camera at the moment, just for the sake of broadband, and start the talk. So I'm going to talk to you today about the ecology and management of badgers and TB. And you'll notice, first of all, that on my title slide, there's a little symbol in the right top hand corner, with a no tweet sign. It's a bit of a joke, really, but I put it there to remind me to tell you that if you do decide to tweet anything that you hear tonight, and it's entirely up to you whether you did or not, then you could well end up in a bit of a Twitter spat with people, because this is a subject that it cites the emotions of people, and there are many entrenched positions, and as we'll find out, the situation is seldom as simple as people often make out. So beware spouting opinions about this complex and controversial issue. So what we're talking about here is an organism called Mycobacterium bovis. It's a bacteria, and it's closely related to the organism that causes TB in humans. It has a very wide host range, which includes a whole range of different mammals. Here you can see pictures of several of the British mammals that Mycobacterium bovis has found to be able to infect. And of course, there are a whole host of other species across the globe that the infection has been found in. And of course, as the name suggests, it also infects bovines. Importantly, and one of the reasons why we are concerned about this disease is it's zoonotic. So that means that it can be transferred between humans and animals. In the picture here, you can see an unfortunate fellow who's got a condition called scrofula. Now, this is the inflammation of an infection of lymph nodes in the head and the neck as a result of drinking infected milk. And this kind of condition in the human population was really quite common before pasteurization was introduced and when Mycobacterium was quite rife in the cattle population. So, bovine TB in cattle. It's a serious disease of UK cattle. It was once very widespread, what we call endemic. So it was throughout the cattle herd across the whole of Great Britain. But since the 1950s, when a rigorous test and slaughter program was introduced, the incidence started to decrease. But sadly, since the 1970s, there's been a resurgence in the incidence of disease in cattle, and it's now a persistent infection in certain parts of the UK. So this figure here just shows you graphically the sort of extent of the increase in the incidence of infection in the cattle population from 86 until 2010, and you can see the number of red dots has increased. Each one of those is a cattle herd infected with TB, and their geographic range has also increased. Fortunately, after about 2010, the incidence of infection started to plateau, and it stayed relatively stable until about 2017. Thereafter, it's tended to decline year on year, but the declines are fairly modest, and the extent of infection in the British cattle herd is still very, very high. And this has a significant cost to government, because government, of course, foots the bill for all of the testing, all of the operational support around that, the regulatory framework, and providing compensation to farmers when they suffer from a breakdown. And of course, it's got a significant cost for individual farmers, and it's estimated that each new breakdown, that's a new incidence of TB in a cattle herd, can cost the farmer around about £19,000. So it's a really significant economic drain. So how do we try to control TB through doing things to the cattle population? Well, as I mentioned before, since the 1950s, we've had a routine testing regime. We are skin testing cattle, and then any cattle that are positive to that skin test, they're removed from the herd. There's also a backup surveillance of slaughterhouse carcasses, so cattle that go through the system end up in the slaughterhouse. They're examined to see if they have lesions indicative of Mycobacterium bovis infection. There are movement restrictions if you miss any testing events, and there's testing of in-contact herds if your herd goes positive. And then there are pre and post-movement tests that are carried out for animals that are moving from the higher risk herds. And more recently, we've had the rollout of a more sensitive test than the skin test, something called the gamma interferon test, and that's extending in use at the moment. And finally, in order to get compliance from the farming community, there's a compensation regime in place. So farmers that suffer from a breakdown, they can be financially compensated relative to the number of reactors that they experience. And those different approaches, different regulations, and so on, they're employed in different ways in the different risk areas in England. So you can see the red area on the left in the south and the west of England. This is the highest risk area. This is where infection is essentially endemic in cattle. And then the lowest risk area is in blue. And obviously the regulatory restrictions are less severe in the blue low risk area. In between the two areas is something called the edge area, and this is where the incidence of TB is changing, sometimes spreading out from the high risk area, and sometimes more recently actually contracting back. So what about badgers and bovine TB? So the first evidence implicating European badgers in transmission of TB to cattle came about in the 1970s, when dead and dying badgers were found on farms that had persistent problems with TB in their herds. Since then, badgers have been culled under a variety of different strategies in order to try to control transmission to cattle. And of course, that method of trying to manage the disease is highly controversial, and there are many animal rights and conservation groups who are very much opposed to that approach. And the eagle-eyed amongst you may have noticed the celebrity rock guitarist Brian May in that photo in the middle there, and he's been very vocal in his opposition to badger culling. But after widespread rollout of culling across the country, and I'll talk about that a little bit later, it's now recognised by DEFRA that the exit strategy from culling will be badger vaccination. And so in years to come, we are likely to see more badger vaccination and less badger culling. And the third tool that we have really to try to manage this disease is to try to manage interactions between cattle and badgers, whether they're direct or indirect, in order to reduce transmission opportunities. And I'll speak about that a little later as well. So TB in badgers, how does it manifest itself? Well, the transmission routes amongst badgers appear to be largely aerosol. So you can imagine badgers living underground in groups, in confined spaces for a lot of time. Aerosol transmission would be quite an efficient way for this organism to transfer from one host to another. And you can see some badger lungs on the right-hand side of those images there, heavily infected with bacteria. But another means of transmitting the disease amongst badgers is when they bite one another. So a badger with infectious bacteria in its mouth might bite another animal and thereby inoculate it with the bacteria. So the kind of pathology that we see in badgers, it's largely a lung infection, but it also affects the lymph nodes of the head and the neck. And you can see in the two bottom pictures there, an animal or two animals with submandibular lymph node infections. The one on the left has burst, and that will be excreting bacteria into the environment. Infected badgers, however, contrary to what some people believe, can actually live for many years, and they can reproduce successfully whilst they're infected and also whilst they're excreting bacteria into the environment. And that's one of the reasons why they're a good maintenance host. Really, they're quite effective at maintaining the disease and then passing it on. And they can excrete the disease, the bacteria in their sputum, in their urine, if the kidneys are infected, also in the feces. During the course of our research on this disease, we found sex-related variation in how animals how they experience the disease. So we find that male badgers tend to cope less well with infection once it's established than female badgers, for instance. We also find behavioural correlates, and I'll discuss some of the examples of that in a moment. So, do badgers give TB to cattle? Well, the pathology and the duration of infection that we see in badgers is certainly consistent with that happening. We've seen associations between sick badgers and infected farms. So there is a correlation, a spatial correlation there. And there have been experimental studies in captivity in the 1980s. Infected badgers were put in an enclosure with cattle, and the cattle were found to then become infected. But that's a very artificial situation. The results of culling trials, where we've measured the amount of TB in cattle in areas where badgers have been removed versus areas where they haven't, suggests that badgers have a role to play, because their removal does certainly change the incidence of TB in cattle. But also there's very convincing genetic evidence that the two species are involved in the transfer of this bacteria. So these are phylogenetic trees or extracts from a phylogenetic tree that show the relationship between Mycobacterium bovis isolates that were obtained from badgers and from cattle living in more or less the same area and at similar times. And where they're clustered along the same vertical line, that means they are almost identical in terms of their genetic fingerprints. And where they're only small distances apart on the horizontal plane, that indicates that they're very closely related. And so from examining these kind of phylogenetic trees, we can see that there is certainly evidence that badgers appear to be transmitting to cattle, but also, interestingly, evidence that at a lesser level of magnitude, the cattle are infecting badgers. So the disease is circulating within cattle and within badgers and being transmitted between the two species in both directions. So what are the routes of transmission? Well, direct contact between badgers and cattle could be a possibility. But we find that when we've done studies looking at interactions between badgers and cattle, they very seldom come into very close direct contact. You might occasionally get situations like this where a cow inspects a dead or dying badger, but more often than not, the two species tend to try to avoid each other. Contamination of pasture by badgers seems to be a much more likely route, where cattle that are grazing in the vicinity of badger feces and urine might become infected. Possibility that badgers and cattle come into much closer sort of artificial contact with one another as a result of badgers going into farm buildings, and I'll discuss that in a moment. So first of all, I'd like to talk a little bit about a study that we've been carrying out, a long-term study that we've been working on in Woodchester Park. This is in Gloucestershire, and the study there started in 1976. I wasn't around then. I joined in about 1996. And the study has involved studying the badger population in a wooded valley, which is owned by the National Trust and is surrounded by private farmland. The area is about 11 square kilometres in total, but the core area where we catch the badgers is about 7 square kilometres. And it's a TB hotspot for cattle. So cattle herds in the immediate vicinity of the study area have quite regularly broken down with TB. And it's also, or it has been historically, an area of really quite high badger density. So the study that I've been involved in, and that's been going since the 1970s, really involves the capture of badgers in the study area and their subsequent examination. So animals are captured in these cage traps that you could see in the top left-hand corner there. They're baited with peanuts, and when they're captured, they're taken back to a facility, anaesthetized, and under the influence of the anesthetic, we test them for TB and take a whole host of other measurements, including obviously recording their sex and their age, but also their reproductive status and various other things. Each animal is given an individual unique mark, so it's given a tattoo on its belly, and it will be released at exactly the same position where it was captured. And hopefully it will be recaptured the next time we trap in that location or some other nearby location. And so as a result of doing that for many years, we've built up a really unique database of over 16,000 capture events of badgers, and that represents over 3,100 different individual badgers. And of course, because some of these animals, or in fact most of them, have been captured many times during their lifetime, we've managed to put together life history data. So you get a series of pieces of information during the life course of individual animals. That's an incredible resource for all kinds of research. Now, principally, one of the most important things we're interested in is the status, the TB status of each of the animals. And so some of the samples that we take are sent to the laboratory to see if we can actually grow the bacteria. And here you can see a Petri dish with some colonies of bacteria growing on it. So we send things like feces samples, urine, aspirates of the esophagus and the trachea, and also swabs from bite wounds and open abscesses. But we also carry out blood tests. So we do antibody tests. So that tells us whether the animal has been exposed to this bacteria at some point in its life. And we carry out an interferon gamma test, which is basically searching for signs of this early cellular response to infection. Now, none of these tests is perfect. They all have limitations in terms of what they can tell us and their performance in detecting the infection. And so what we have to do is we have to combine all of that information in some way. We combine it, and then we get out the other end some idea of the probability that an animal was infected at a particular point in time. So rather than a sort of yes, no binary, yes, you're infected, no, you're not kind of conclusion, we're able to say whether we have a 70, 80, 90 or whatever percent probability that the animal was infected, given the information. And then you can build up a picture of the pattern of infection in the population over time. At our best, one of our best guesses of what that looks like between 1982 and 2010 looks like this. And you can see that the prevalence, so the proportion of the population of badgers that's infected at any point in time, is generally lower than about 15%. Occasionally, especially after about 1995, occasionally peaking higher than that. And it seems to oscillate over time. So obviously, when you see those oscillations, you start to wonder, well, what's driving changes in disease prevalence in our population? And this is a really fundamental question for us. One of the first things that you would look at in that situation is the density of the host. So in many diseases, we have what's called density-dependent transmission. And so you expect in those situations for transmission to be greater where the density of hosts is highest. Now, in this plot, you can see over many years the numbers of badgers captured in our study area, numbers of adult badgers captured. And that's essentially a proxy for density in our study area. And then overlaid on those bars is a black line, which indicates the percentage of incident cases. So these are new cases of disease in our badger population. And you can see that there's no simple relationship between those two things. So it doesn't seem that density is the primary driver of the incidence of new cases in our population. So what's driving disease in this wild animal population? Well, we think that something that might be important in driving disease may be the spatial structure of the population, because badgers aren't just homogeneously mixing together in the population. There's actually some structure to it. Badgers live in social groups, and those social groups tend to be territorial. So you can see these stars here representing main sets, main breeding sets, which are the focus of the activity of a social group of badgers, and then a polygon around them which represents their group territory. And they're fairly distinct, and they give the population spatial structure. And so that limits the amount of movement between groups. So the population, it's not randomly mixing. In fact, most animals are only having contact with their immediate neighbours. And so this tends to limit transmission. And it means you don't get density-dependent transmission. Transmission patterns are much likely to be a little bit more complex than that. And we certainly see that configuration of social structure limits disease transmission when we look at the distribution of infection in the population. So this is a diagram indicating the distribution of social groups. So each of those circles is a social group of badgers, and the size of the circle indicates the number of animals in that social group. The polygons represent the territories of that group. And just for the sake of this talk, if you look at where the red and the yellow sections are, those indicate infected animals. And you can see that they're confined to just a handful of groups. And although this is just an image for one of the years between 1982 and 1996, when we carried out this analysis, all of the other years looked pretty similar. So it appeared that the infection was staying really quite stable in our spatially structured population. However, some badgers do decide to move around. They do move between social groups. And in this graph, you can see a pink line, which represents the proportion of the population that moved between social groups each year. It never really rises above 16%, so it's a minority of animals, but some do move every year. And then the line below it, the blue line, is the incidence of new infections again. And you'll have to take my word for it, but those two lines are significantly statistically associated with one another, such that years of high movement are followed by years of increased incidence in the population. So that seems to suggest that movement may be important in driving incidence in the subsequent year. So we looked in more detail at this, and we looked at individuals that moved. And we found that individuals that moved had an increased risk themselves of becoming infected, but also groups that experienced more movement, whether it was into the group or out of the group, they also had an increased likelihood of new cases arising. So this was really very strong evidence that movement between groups is a really important driver of transmission in our system. So why is that important in terms of management, for example? Well, it's important because it relates to something called social perturbation. And this is the spatial disruption of badger social groups that are subjected to culling. You can see in this figure here, on the left-hand side, you see an undisturbed population. Those polygons are the social group territories, and they're all sort of aligned together, abutting against one another. But then in the culled population, there's a lot of overlap between those group territories. Animals are wandering around much more, and that effect bleeds out into the neighbouring unculled, undisturbed population. So you get quite a lot of disruption at the edge of a cull area. And in those disrupted areas where you've got these overlapping home ranges, then there are enhanced rates of movement between groups, and there's more opportunity for transmission. So although when you're culling, for example, you're reducing the density of the population, those animals that remain, they are moving around more. It's providing more opportunity for transmission, and that probably explains why culling can have positive and negative effects, which we'll talk about in a moment. So just before moving on to look at the three different management approaches, a couple of other pieces of work that we've done that relate to that illustrate really how understanding the ecology of the animal is so important in understanding how to manage the disease. The first is radio tracking. We do an awful lot of radio tracking in our study area, and in one study we looked at how animals use those main breeding sets largely towards the centre of their territories, as opposed to smaller outlying sets in their territories. And what we found was that badgers that were TB positive, according to our tests, you can see on the left-hand side in this graph, tended to spend more time away from the main set. And this is a seasonal effect for all badgers. So most badgers spend less time around the main set during the summer and more time around the main set in the winter. But you can see from that plot that it's a really exaggerated situation in those animals that are positive. Now that's really important and has implications for management, because if for some reason you want to make sure that you're targeting those positive animals in terms of management, then you need to understand that they're not likely to be in the main sets a lot of the time during the summer. Another piece of work that we've done that relates to management is looking at social networks in badgers. So we're able to deploy radio collars, as I said before, but we can also deploy these collars, which are proximity loggers. And these will log the presence of another collar. So if you stick enough of these on the badgers in your population, then they will download information on contacts between individual animals. And you can build a social network like this. And this has been really interesting, looking at the test positive badgers in the population and the test negative animals. And what we see is a distinct difference in such that the positive animals, they tend to be less interested in connecting with members of their own social groups, but they have quite strong links with animals from other groups. And if you just look at that big red square in the middle of that social network there, it's very well connected to a number of other clusters of individual animals. And what this means is that these animals can be really pivotal in terms of spreading disease. So again, from a management point of view, it's really important to understand whether some individuals might be more important for disease spread than others. And this relates to the epidemiological phenomenon of super spreaders. Okay, so we'll talk now about the three different sort of broad management approaches. The first thing I'll talk about is badger culling, and As I said before, since the 1970s, there have been several different approaches to try to use culling to reduce transmission opportunities and reduce infection in cattle. In 1997, culling was suspended and an independent government review reported. And that review, chaired by John Krebs, concluded that a randomized badger culling trial was required in order to work out whether badger culling could actually have a positive impact on TB incidence in cattle. And this piece of work has provided a lot of important evidence in our understanding of the impact of culling on badgers. So in the trial, they compared TB in cattle herds in areas that were subject to culling against areas where there was no culling. And these areas were grouped in groups of 3 called triplets. In the green circle areas, no badger culling took place. In the yellow areas, what was called proactive culling took place, so badgers were culled across the entire area. And in the red areas, the reactive areas, badgers were only culled in the immediate vicinity of farms that had a TB breakdown. And you can see the distribution of those throughout the infected areas of England. Now, the results of the RBCT were very, very interesting. They showed us that culling can have positive but also negative effects on TB in cattle. So the reactive culling, so that was just culling around farms where you'd had a breakdown, actually resulted in a 27% increase in TB in the cattle. And so that treatment was stopped early by ministers. In the proactive area, however, there was a 25% decrease in cattle TB in those intensively culled areas. But if you looked around the edges of those areas, you found that cattle TB actually increased. And the magnitude of that increase meant that overall there was no benefit from the proactive culling during the period of culling. We also saw during that period of culling that the incidence of, or rather the prevalence of TB in badgers actually increased. That's supporting this idea that where you mix up, where you stir up badger populations, you actually increase opportunities for transmission. But interestingly, the negative effect of culling on cattle TB actually disappeared after the culling stopped. And the benefits, however, accrued. So it transpired that during the years immediately after culling, there was actually a benefit in terms of TB incidence in cattle. I think it's fair to say that the overall benefits of culling are really modest compared to the amount of effort that's required. And the reason why they're so modest is probably the effects of this perturbation phenomenon, which means that when you're culling, you might be getting some benefit from reducing the amount of badgers around, but you're also creating a situation where you get some of these counterproductive effects as well. So the current culling policy started in 2013 with 2 areas in Gloucester and Somerset, and then gradual rollout to many other areas. The number of active cull areas in 2022 was 72, and you can see their distribution on this map here. So that is really quite a huge number of areas involved in culling. The approach was very different from what was taking place in the randomized badger culling trial. It was led by farmers. That's the most important difference. Badgers were not only trapped, but they were also shot. The culls went on for 6 weeks with some extensions possible, and there were various requirements to cull areas being given a license, which related to trying to reduce this perturbation effect. So to date, there have only been limited analyses of that data. There will be more coming out shortly, but the analyses that have been done tried to look at changes in TB incidence rates in the cattle in 3 of those areas relative to comparison areas that weren't culled. Now, after 2 years of culling in Gloucester and Somerset culling areas, there were quite modest decreases in the amount of TB in cattle in those areas compared to unculled areas. There was no change in a 3rd area in Dorset. But in that culling area, after 4 years of culling, the reduction in TB in the cattle was really far more substantial in Gloucester and in Somerset, but too early to tell really still in Dorset. So what can we conclude from all of that? Does badger culling reduce TB in cattle? Well, the evidence from the culling, from the current culls, certainly suggests that they can reduce TB in cattle, but we can't necessarily say that they always will, because it's a very small number of areas that have been looked at so far, and the results might differ quite a lot in other areas, depending on the effectiveness of the cull, depending on the size of the badger population, depending on the cattle population. Also, it's possible that other factors may have had an impact as well as culling in this small number of areas, because of course, you know, there has been a rollout of other measures to try to reduce TB in cattle alongside culling. We don't know what the longer term impacts would be. That small scale analysis that's been done only looks at 4 years. We don't know what would happen for longer. Many of these cull areas may go for 6 years of culling. So we don't know the longer term impacts, and we haven't seen anywhere yet any reductions of 100%. So that certainly means that we need a combination of measures to really get on top of managing this disease. So let's talk a little bit about one of those other approaches, which is badger vaccination. The first thing to say is that the vaccine that we're using here is the Bacillus of Calmette and Guerin, and this is a vaccine that was developed for use in humans, and it is the mainstay of TB control in the human population across the world. There are 2 delivery options for vaccines. The first is to inject it into captured badgers. We've carried out some studies that show that it's safe. We've showed that it does not prevent infection in badgers, but it will reduce the severity of infection in badgers, and that could be enough to reduce the amount of transmission. So those are results from studies on captive animals. You could always also incorporate the vaccine into a bait, which is then distributed and the badgers consume it. The efficacy of that approach has been demonstrated in principle on captive animals, but it is very technically challenging to develop this formulation of a bait that's a happy medium for the live vaccine to live in, that badgers also like to eat, and that will also persist in the environment for a while. So concentrating on the injectable vaccine, we carried out a field study in Gloucestershire to look at what happens when you vaccinate badgers in the wild. And what we were able to find was that injecting them with BCG significantly reduced the risk of a positive test result by between 54 and 85%, depending on which test combination you used. And this corresponded with the results from the captive studies. So that was all very encouraging, showing a direct positive effect of vaccination in badgers. What it also showed was that there was no effect if you stuck the vaccine into an animal that was already infected. But interestingly, what we also discovered, which was a little unexpected, was a beneficial indirect effect. So this showed that you could significantly reduce the risk of a positive result in a cub when more of its group members were vaccinated. So what this is a reflection of what's called the herd immunity effect. And we'll all be familiar with that term from COVID over the last few years. But essentially, it's the indirect benefit that unvaccinated animals get from the fact that you're simply reducing the amount of infection in the whole population. So how has deployment of the vaccine gone over the last few years? Well, the Badger BCG formulation was licensed for use in badgers in 2010. And since then, it's been deployed by a variety of organisations. There's been a gradual increase in the numbers of badgers that have been vaccinated year on year. In 2021, 1,575 badgers were vaccinated. It's still relatively small scale, also small scale in terms of geographic distribution, but it's starting to get some real traction. And as DEFRA is now investing heavily in DEFRA-led vaccination in post-cull areas, so encouraging these cull companies that have come to the end of their culling period to take on vaccination now, we might see much wider deployment of vaccination. But one of the important challenges in relation to that is to engage better with the farming community and to encourage uptake of vaccination after culling. And it's I think it's fair to say that there is still a certain amount of skepticism about badger vaccination in the farming community. But one important piece of evidence that we don't have, we don't know what the effects of vaccinating badgers are on TB in cattle, although it stands to reason that if you're reducing infection in the badger population and they are in any way connected with TB in cattle, that you can only have a positive effect on the latter. So how many badgers might we expect, or what proportion of the population might we expect to be able to vaccinate by trapping and injecting? So we carried out a study in the context of a vaccination project in Wales, which was where they attempted to vaccinate badgers over 288 square kilometres. In the 4th year of their vaccination, we collected badger hairs from badgers that were being vaccinated, but also from hair traps that we put out in the wider area. And then we examined the genotypes that we got from the hair samples. And by doing that, we were able to relate the subsample of animals that were trapped and vaccinated to the wider population and get an estimate of vaccine coverage. And what that showed us was that you could expect in the first year of a badger vaccination program to probably hit about 50% of the animals in that population, which doesn't sound terribly encouraging. But if you look at what happens in subsequent years, we modeled what would happen over the course of 4 years. By the 4th year, you're hitting, somewhere between almost 70 and 80% of the badger population. And that's a much more encouraging level of vaccine coverage. So the final management sort of approach that I'd like to discuss is managing interactions between badgers and cattle. The first thing to notice in this picture is that that is not a real badger. That is a stuffed badger. We had to stage this photograph because direct contacts between badgers and cattle out on pasture are extremely rare. Indirect contact is far more likely. So cattle coming into contact with badger feces and urine on pasture. There might be hotspots for contamination, like cattle feed troughs, also buildings if badgers are attracted to them. And there's even possibilities of direct contact in buildings. So we've had a number of studies where we've looked at badger visits to buildings because that's clearly something that farmers can definitely do something about. And what we found was that when we looked at about 200 farms where we carried out video surveillance in their farm buildings, we found that the level of visits could be quite common. In fact, 39% of our 200 farms experienced some visits to the buildings. And on some of those farms, visits were occurring really regularly, almost every night. And in those situations, there were far more visits during dry weather. And this is an interesting phenomenon. And you'll see from the graph here that the number of badgers that we were likely to see in our video surveillance footage was greatest when the rainfall in the previous 24 hours had been low. And this is probably because when rainfall's high, badgers are out on the pasture hoovering up all the surface feeding worms. But when rainfall is low, then the conditions are dry and those earthworms are not likely to come out of their burrows and feed on the surface. And so badgers not fancying digging for all their worms are likely to go into the farm buildings and take feed from easy pickings from the buildings instead. So that provides opportunities for indirect transmission, contaminating the environment, but also potentially for very close contact with cattle, unlike out on pasture. So I'm really hoping this is going to work. This video, I hope it looks like it's playing. You'll see this is a bull that's penned up in a farm building, and the badger is entering from the right there and moseying around. Neither of them seem too bothered by the presence of one another. In fact, there's a little bit of a sniff, nose to nose there, quite close. And what this footage tells me really is that these two are no strangers to one another. I think this has probably happened before. So clearly an opportunity for transmission of disease, not only, of course, from badgers to cattle, but also from an infected cow to a badger. This next video is just a little bit of fun, really, but it does illustrate how badgers can become very habituated to feed that's available in feed sheds. You'll see a whole host of animals on the right-hand side of the screen there, illuminated by the camera. And they're feeding on some spilt cattle pellets. So there's a bag there that's split, and they're feeding there. There are also bags of food on top of that pallet. And in a moment, look, they all look up. Suddenly they're a bit agitated. That's because there's a farm dog outside barking. They're all wondering what's going on. And then they've all had enough and they rush off, and there's a whole bunch come out from under the pallet. So I think if you're quick enough, you can count about 11 or 12 badgers in that footage. So how do we manage risks from those kinds of situations? Well, if you look across the bottom of the screen there, you can see some simple preventative barriers that can be put in place to help secure farmyards, buildings, and feed on farms. And these measures are simple, and we've shown them in trials to be 100% effective, as long as you do remember to shut that gate or pull that roller door down or whatever. So if in the process of running your farm you forget to close the gate, the badgers are straight in there if they're used to going in there already. We've also looked at a farm assessment tool. So this is a way of trying to predict the farms that are likely to get badger visits, because putting those kind of measures in place costs money, and you can only expect farmers to make informed decisions if they've got good reason to believe that their farm is vulnerable to badger visits. So I'd just like to end with a slide on future research. These are areas, hot topics of future research. These might be things you want to ask me questions about when we break out into the discussion. Things that I'm particularly interested in following up in the future, and I think the department is also interested in. So things like what creates new foci of infection, what's the effect of vaccinating badgers on cattle, how do we identify and remove infected badgers as opposed to any old badger, and can we identify those farms that are most at risk of, for example, badgers getting into the buildings. So finally, just going back to my original comments about inaccurate information flying around about this subject, there is an awful lot of nonsense out there, a lot of cherry-picked evidence and so on. If you want some good objective information, some good evidence summaries, then try these two websites. They're very reliable, very objectively presented information available. And it just remains for me to say thank you very much for your attention. Over to you, Debbie.
Thanks very much, Des. That was absolutely fascinating. We have got some questions in the chat. I'll just read them out. Rosine Black asked, how quickly do the results take before badgers are released? This is going back to when you're capturing badgers, doing the blood tests, etc.
Yeah, good question. So when we're doing our routine scientific research at Woodchester, the animals are only kept overnight and they're released the next morning. That's on the first day of trapping, and on the second day the animals are caught and then they're released on the same day. So they're only ever kept for overnight at the maximum. We send all the samples off to have the tests carried out. We don't wait for the test results to come back before we let the animals go. And in our project, we have known infected animals that we are catching, sampling, and releasing. So we are releasing back into the wild known infected animals. But that's the purpose of the study. The study is to not intervene. It's to look at what happens in a badger population that's just left to its own devices. There are a couple of tests available, one in particular that you could use and get a result in 5 minutes next to the trap, or 5 to 10 minutes, and you could get a result there and then. So those kind of tests could be used if you were going to take some action on the basis of a test result.
For example, if you were going to take out an infected badger.
Correct, yeah. The problem at the moment is that those tests aren't terribly reliable.
Right. Another question, Ray Smith asks, why aren't cattle vaccinated?
Yeah, another excellent question. The answer is that there is a very active research project being carried out, developing vaccines for use in cattle. The same vaccine that we've used in badgers has been shown to have some positive effects in cattle in trials. And it's likely that we will see field trials of vaccination of cattle starting sometime soon in England. One of the previous barriers has been to do with markets, basically, and selling cattle products onto other markets where the competition is not vaccinating their cattle, and so it could be deemed as a disadvantage to be vaccinating your cattle in some respect. So there's been a lot of talk around this and around whether Brexit has actually increased the chances of getting rid of that problem. I'm no expert on that particular situation, but I know that there is very active research. We will see cattle vaccines being rolled out in field trials quite soon, and I'm hopeful that we will see them deployed more widely in the future.
Good. Rosen is asking again, why might a farmer choose to vaccinate badgers? How affordable is it in time and money? It seems very labour intensive.
It is quite labour intensive. You know, you have to catch the animals in order to vaccinate them. The organisations that are doing vaccination are either government or wildlife trusts and other wildlife organisations. Some of those voluntary organisations have approached farmers to ask if they would like them to vaccinate animals on their land. And government vaccination teams have been vaccinating on farmers' land as well. So government is very keen to carry out vaccination, especially in areas that have been subject to culls in the past. So there are ways and means of at least some farmers in some parts of the country accessing a lot of support for getting their badgers vaccinated.
Right, Des, we've also had a request for you to go back in your slides so that you're showing the two reputable websites again.
There we are.
That's it. Okay, hopefully that means people can copy them down. Now, Drew is asking, what can we as a community do to help eliminate misinformation?
Yeah, that's a good challenge for us, isn't it? Well, I think as sort of many of those who are listening are professional ecologists. You know, I think our views carry a certain amount of credibility in certain circles, and so you know we should familiarise ourselves with the bare bones of the evidence. I think so that we can present an objective point of view. I think especially those of us who go out and work on badgers in the context of consultancy work should understand this subject a bit more. By promoting sites like this, I think we can promote, you know, direct people to good sources of evidence. Don't get involved in the pointless spats that go on Twitter and so on would be my recommendation. Just point people to these sources of objective information.
Right, well, there are no more questions. There is one coming in, but I'm just going to get my question in first. Yes, cattle, humans and badgers can all be affected by TB.
Yeah.
What about other animals? What about other wildlife?
Yeah, so if we just look at the UK, for example, we did a study some years ago now where we sampled a whole range of British mammals. And we found really that, you know, the harder you look, the more you find. So we found infection in a field vole, we found it in a grey squirrel, we found it in foxes, various other species, most of which are probably not very important in terms of transmission, because they're probably not maintaining the infection like badgers do. The one exception to that is deer. So deer in some areas where they're of sufficiently high density to maintain the infection, they could be important as a source of infection for cattle. And potentially in some areas, there might be circulation between cattle, badgers and deer going on. So, but I think all of the evidence really points out badgers as being the most important wildlife host in this country. In other countries, other species are important. Buffalo are affected in South Africa, for example. Lions are infected in South Africa, and that's a big problem for conservation because lions and other big carnivores are a big draw for the national parks, and there's significant, you know, revenue to be made out of the tourism associated with that, and it can be quite decimating to felid populations. In Australia, they've done very well in eliminating infection in a lot of wildlife hosts. In New Zealand, they've also managed to start to really stamp out infection in possums, which were the wildlife host over there.
Thank you. Now, just a last question, because we really are running out of time, because we do aim to stop dead on 7. Lee is asking, what's your view on the best management approach based on research to date? A combination of all 3, cull vaccination and farm management, or vaccination and farm management? And he says, obviously, as an ecologist, culling is a last resort consideration.
Yeah, well, I think that's a very understandable position, and you know, from my point of view, I think I would say that trying to manage interactions between badgers and cattle has got to be the first line of defence. You know, if the most important route of transmission on a farm is badgers getting into a feed shed, then preventing them from getting in has got to be the first line of defence, hasn't it? Obviously, there are ethical reasons to prefer vaccination to culling, and you know, applying culling in a very widespread way, in a very non-discriminatory way, you know, just against, you know, you're culling all of the badgers regardless of their infection status. You know, there are arguments about whether that is proportionate. It's an awful lot of effort. There are ethical concerns around it, welfare concerns, etc. So I can see why vaccination is a much preferred option. I would like it if we could roll out vaccination much more widely, roll out much more management, understand better what the causes of infection in cattle are on individual farms and on clusters of farms, so that we can tailor the management and control techniques to the particular situation. I think that's a key area for more work in the future. But I think that culling will sadly have a place, hopefully a much smaller place than it has in the past, but it will have a place in stamping out disease where everything else seems to fail.
Thank you, Des. Des, could you stop sharing your slides, please, so that I can share my screen for the wrap up, just so that we can mention what's coming up next. We do have, I don't know how many of you are members of CIEEM. We have these meetings are open to non-members as well as members. But if you are a member and you've signed up to be a member of the ASIG, the Academic Special Interest Group section, then we do have our annual members meeting in March instead of having an open meeting, and we have Kenny Cortland talking about the Cairngorms Connect Predator Project. If you are a member of CIEEM but you haven't signed up to the Academic Special Interest Group, you just do it. You just sign in and you update your personal preferences and sign up to the Special Interest Group, the academia one, and then you'll get news about all our events. And you can come along to that members meeting and share your views on what we should be doing. What would you like to hear talks about? Is there anything else you'd like us to be doing to facilitate this bridging between research and practice? With that, I'd just like to say thank you very much, Des, again. And I suspect other people in the chat are saying excellent talk, brilliant talk, thanks very much. But with that, it is dead on 7 o'clock. So thank you very much. And you can all leave now and go and have your supper. Drew's put up the link to the March event, but please note it is only open to CIEEM members. It's just once a year we have a closed meeting. Thank you very much. Hope to see you all again at another event.